Welcome back, everyone. So we are continuing on with our topic 2, the slippery slope of life. So we are on to part 5, what are viruses? So now that we know the components of extracellular or eukaryotic cells, we're going to take a look at some things that we're probably familiar with that are actually not considered cells, and we're going to discover why they're not considered cells. So why are viruses not considered cells? So by the end of this lesson, you should be able to describe the size and structure of a virus, as well as identify how viruses attach and enter the cells they infect. So let's start out with talking about, well, what are viruses? So viruses, they are acellular, okay? So first off, acellular. Cellular means cells, so acellular means without. So they are not considered cells. And they're considered parasitic because they will invade a cell and take it over, okay? So they're parasites. So let's go ahead and figure out, why are they not considered cells? Well, they're not considered cells because they don't have all components of a cell. So if you think about the basic components of a cell, there's 4 of them, right? We talked about that earlier in topic 2. All cells have a plasma membrane. Well, the viruses don't have a plasma membrane, so that's one disqualification right there. All cells also have ribosomes. Viruses don't have ribosomes. All cells have genetic information, DNA or RNA. Well, viruses do have genetic information. They do have DNA or RNA. So that's one that the viruses do have. And then all cells have cytoplasm. Viruses don't really have that. So viruses, they have one of the 4 components of a cell. So that's why they're not considered alive. And then if we think about the characteristics of life, a virus does not have all the characteristics of life. So for example, they don't have metabolic processes. They can't take one form of energy and convert it to another like our cells can do. They don't divide like our cells. So they actually have to infect one of our cells and then trick our cell into taking the virus's DNA and dividing it and make more viruses. So they don't have the same, they don't have all of the characteristics of life. So viruses are not considered living things. Okay? But viruses, even though they're not considered alive, they can infect all forms of life. So that includes bacterial cells, that includes plants, that can include fungi. It includes, of course, us humans, because, if you've ever had a cold before, that's a virus. And viruses are very diverse, okay? So, there's a whole host of different viruses out there. And, they do different things. So some of them can be very harmless, pretty harmless, like the simple cold that kind of is bothersome, but it's not going to really hurt you in any long-term effect, to viruses that can potentially kill people. So they are very diverse. So since viruses, they are not considered living, well, how do they replicate? Well, first of all, let's take a look at their size. Okay, so let's take a look. These are very, very small viruses. So if we look at our size chart here, so just to review, remember bacterial cells or prokaryotes, they range from size between about 1 micrometer to about 5 micrometers. Okay, so they're right around this area of the scale, about here to here. Whereas our eukaryotic cells are about, about 10 micrometers to 100 micrometers on average. So about here to here. Viruses are a lot smaller. Okay, so If we just look at one single virus cell particle, it's called a viron, and they are anywhere from 20 to 250 nanometers. So remember, a micrometer is 10 to the negative 6. Okay, so that's, 1 divided by, 1 was 6 zeros, so 100,000. But nano means 10 to the negative 9. So that's a lot smaller. So 1 over, 1, 9. So that's, a lot different. So nanometers are very, very small. So we're talking, very, very small, a lot smaller than a bacterial cell. So we can't even see them with a light microscope. Okay, you would only be able to see them with an electron microscope. And so these images right here, you would have to see, these are from, guess what, an electron microscope. These cannot be seen with a light microscope. Okay, and as far as how we classify them by basically, well, we used to classify them strictly based on what type of DNA or RNA they had. So basically, we classified them if they housed DNA or RNA. So some of them were RNA type viruses, some of them were DNA type viruses. So based on what they carried, do they carry DNA or RNA? But more recently, we take a look at how they replicate. So their viral replication cycles. So that's how we're beginning to classify them based on that. All right, so let's take a closer look at how exactly they classify. So a viron, as far as what they are made of, we already know that basically the only thing that they contain that is similar to a cell is DNA or RNA. So they do have that in their, in the core of their, can we even call it a body? A body? I don't know. So that's housed in the middle. So that is either a DNA or an RNA strand. And then surrounding that is a protein coat. So that's called a capsid. Okay. And so that surrounds the genetic material. And then some of them have another coat, which we call the capsule, okay? So the envelope. So the viral envelope can be on the outside of the capsule, or the capsid, excuse me. So, so basically you got DNA in the middle, a capsid, and then you might have an envelope surrounding it. So, so that's basically what it is. All of them have glycoprotein. So glycoprotein, glyco means like a carbohydrate, a sugar, and then protein is protein. So all of them have glycoprotein that kind of sticks out of their capsid, and that's what they use to attach to a cell. So cells on their plasma membranes, they have glycoproteins. And so that way they can kind of match up with a host cell that way. So they can, match up to a receptor cell site that way and possibly infect that cell. And just by looking at this picture, you can kind of see the different types. They look really weird. Like, look at this guy, this bacteriophage. So viruses, they can be pretty complex in shape. So we do have, some weird ones. This adenovirus, you can see inside, and then you have, instead of a capsid, you got these little protein units, which are called capsospheres, that kind of make up this capsid. HIV, you've probably heard of HIV. So here's what it looks like, and you can see inside, it has RNA, and then you have the capsid, and then you have the viral envelope, and then you have the glycoprotein. So there's, they're pretty diverse in shape and what they look like in their structure. All right, so let's talk about how they exactly infect a cell. So the first part is they're going to attach to the host cell. So remember, they have those glycoproteins. So here's like an influenza virus, so the flu. The flu is caused by a virus, and so here's our influenza virus. And this is just an example. So the influenza virus has these little glycoproteins that stick out from the capsid, and it's going to attach to a receptor site on the cell membrane of the host cell. Okay, so that's the first step. The virus is going to attach to a specific receptor site on the cell membrane. And the virus is kind of tricking the cell at that point. It's mimicking, like, hey, let me in, I got something for you. And so the virus is going to penetrate the cell membrane and enter the cell. Okay, so it's a specific glycoprotein that's supposed to bond to this specific receptor site. So the cell assumes that, it's supposed to be receiving whatever the package is. And so the virus enters the cell and penetrates the cell membrane. So there it goes, it's entering the cell. And then it's going to uncoat or release its contents. So you can see how it's releasing its contents into the cell. And then the DNA or RNA, depending on what it's carrying, enters the nucleus. So you can see how the DNA or RNA enters the nucleus for replication. So inside the viral messenger RNA, it's going to replicate. So we have the genetic code is replicating, and so that's going to trick the cell to make more viral proteins. So that's the fifth step. The viral messenger RNA is used to assemble viral proteins. So now we have the cell is actually doing the work of the virus to make more viruses. Okay, so new viral particles are then made and then are released into the extracellular fluid. So during this time, the cell is, a lot of times the cell is basically, it's going to self-destruct after this. So the cell is dying in the process. So the cell has been taken over and it's been tricked to create more viruses, it's not a cell. And then the host cell ends up dying in the process. So as far as viruses, viruses can cause a variety of diseases. And as I said before, these can be, pretty harmless diseases like the common cold to potentially fatal diseases like something like meningitis. And you can see, there's a number of viruses that can infect humans. So you can just see some of them you've probably heard of. So like rhinoviruses is a fancy word for the common cold. You've probably heard of rabies. So that's something that's not very common, but can be zoonotic virus, so it can be transmitted from animals. Measles, you've probably heard of that. So some of these can be treated with antiviral drugs or by vaccines. So measles is not really common anymore because we have a vaccine. Let's see, what other, mumps, that's a vaccine that you've probably had, so it's very rare. Influenza, so that's the flu. So influenza, there's a number of different types of flu viruses, and it mutates quite often, same with the rhinovirus. And so that's why we have to get a vaccination every year, because there's so many varieties of it. So basically the CDC or whatever is trying to figure out predicts which one's going to happen this year, and then gives you a shot for it, a vaccine for it. SARS coronavirus, hey, what's that one? Polio is a virus. It was caused by a virus. Let's see, there's another coronavirus that's more gastro-related. Sexually transmitted diseases, so like HPV, so some of you may have been vaccinated against that. Herpes is a virus, HIV. Let's see, smallpox has pretty much been eliminated. Well, it has been eliminated from our population. Rubella is, something that we get as a vaccination when we're kids. Hepatitis, so that some of you may have received vaccinations for that as well. So there's a number of vaccinations that we can take to prevent us from getting particular viruses, and then there's also antivirals that we can take if we do get one of these viruses to prevent us from getting really, really sick. But as we know, especially with our recent experience with the coronavirus, viruses do mutate. So, we saw with coronavirus, we see different mutations of it. And so, that's why they recommended those boosters and so forth. And then they can become resistant to antiviral drugs. So there are some things that can occur as they mutate. Okay. All right, so here's a review question. So which statement is true? Is it A, viruses replicate outside of the cell? B, viruses are acellular? C, a viron contains DNA and RNA? Or D, most viruses are easily visualized with a light microscope? If you chose B, I would agree with you. They are acellular. If you're thinking it's C, remember it's either DNA or RNA. It's not both. And D, they cannot be visualized with a light microscope. They can only be visualized with a electron microscope. All right, so that's it for part 5 of topic 2, what are viruses? So by now you should be able to describe the size and structure of a virus, as well as identify how viruses attach and enter the cells they infect. So for our next video, we're going to look at another acellular being that can infect our body, which is a prion or pyron. Okay, so we'll see you there.